So the first very interesting field we are very excited is about the nudge signaling. So basically, nudge is well conserved signaling molecules, and which during the development and during this tumor growth. And one thing is very interesting is like under different mechanical environment, and actually that's the same nudge pathways. They could have either as a tumor regressor, or they could be a tumor promoter. So they have really opposite effect on the tumor growth. So we feel very excited to see, like, we want to understand how this, based on this attention probe, how really what happens, like what kind of mechanical transduction happens in this environment. So another evidence is like, for example, for this immune cells. And so immune cell for this mechanical transduction has been kind of well studied, but still has many questions. So for example, for the recent Nobel Prize and given to the piezo-1, which is one of the mechanosensitive ion channels on the membrane. And actually that is one of the systems you can use it to use a mechanical force to regulate and to control this kind of immune cell activations or signalings. And similarly, you have this T cell receptor, you have B cell receptor, which is used to regulate how the activation of the T cells. So that could be potentially useful for the immunotherapy. So I think that will be also a very interesting field, how these mechanical sensors can make a contribution to.
Tension sensors are extremely useful for many areas of biology, spanning from like the molecule, cells, and tissue levels. At the molecular level, tension sensors will provide us the opportunities to dissect, interrogate, understand the operating principle of key mechanosensitive proteins orchestrating many neurological, immunological, and developmental, and pathophysiological processes. For example, the tension sensors can be used for studying molecular mechanisms underlying mechanotransduction of piezo channel, the pain-sensing protein, and this year's Nobel Prize winner protein. And tension sensors also provide deeper mechanistic insight of immune receptors and checkpoint inhibitors, such as TCR and PD-1 and CTLA. These are key regulated molecules for immune cell functions against pathogens such as cancer cells, bacteria, and viruses such as coronaviruses. At their cell and tissue levels, these mechanoproteins serve critical regulatory roles in cellular communications. The molecular tension sensors would allow mapping of mechanical and cellular signal networks in multicellular and tissue contexts, and eventually provide systems-level understanding of force and signal transmission in biology during developmental and neurological processes.